Population decline is becoming more prevalent in a vast array of species. Amphibians are the world's most extinct prone vertebrate, with 33% currently threatened. In this study, Dean Newell, R. Golden Gay, and L. Brooks studied a Mixophyes fleayi, a stream-breeding frog, which had undergone population decline after the species was infected with the amphibian chytrid fungus Batrachochytrium dendropatitis, or commonly known as BD. The aim of this study was to characterize the route of the two Mixophyes fleayi populations infected with BD by performing a mark recapture study at two locations in national parks in northern New South Wales over a 7-year period. A frog survey and marking was conducted at night every year from 2000 to 2008 using a head torch and 50-watt spotlight to detect the frogs via their eyeshine, between August and March, because that's when the frogs were intermittently active. The researchers also used call playback and call mimicry to get a response from the males at Turntable Falls. The frogs were called by hand and tagged with a passive integrated transponder and then read with a portable scanner. They tagged 136 frogs at Brindle Creek and 152 at Turntable Falls, and then encountered those individuals 974 times. For data analysis, mark was used to estimate the probabilities of detection and adult survival and to explain changes in population size over time. They needed to consider the number of frogs entering the sites during the study period, whether they survived until the next survey time, and their recruitment level over time. The capture, survival, recruitment, and abundance probabilities were all calculated using the Poppin formulation of Jolly-Seba. Figure 1 shows the comparison of the two locations. The frogs at Brindle Creek survived longer, up to 6 years, than those at Turntable Falls. There was a steady increase in recruitment at Brindle Creek, and even though the recruitment was inconsistent at Turntable Falls, it produced a higher percentage in recruitment. The abundance probabilities represented a 3 to 10-fold increase in abundance over the 7 years. Newell, Golden Gay, and Brooks' hypothesis was supported by the results obtained. The Mixophyes fleayi populations did increase in abundance. Usually the focus of population growth is on increasing survival of eggs and larvae in aquatic habitat. But this study showed the importance of adult survival and their ability to breed. Longevity of adults may be vital in assisting population growth during times of low recruitment. Impacts on reproduction may take many years to recognize a decline in the number of adult frogs if surveys are based on counts of calling males alone. The results were restricted because the analysis didn't include many females, even though the area was breeding habitat. While BD has become endemic, the population has increased to pre-declined numbers. Further research is required to investigate the host-pathogen relationship and discover if environmental conditions, a decrease in transmission or virulence, or the infection, or the possible development of an acquired immunity in the frogs has allowed the increase in population growth. So overall, this study was successful in recovering two populations of Mixophyes fleayi by using the mark recapture method. By surveying the population periodically, they'll be able to monitor the future dynamics of the population.